Okay, guys, welcome back to another anatomy and physiology lecture. Today we're going to be starting on the cardiovascular system and we're going to be going over blood. As always, I've given you a set of learning objectives that I would pay attention to kind of help guide you through the lecture. And there's also a lecture handout that I've uploaded on Blackboard in both a Word and a PDF format. And if you pay attention to the follow alongs, those should help you kind of pace with what we're talking about during the course of the lecture. So pay attention to those, pause the video where you need to, and think about those practice questions that are coming at you in that follow along handout that I've provided for you. So today we're going to be talking about the cardiovascular system with a focus on blood. And I always like to start out a discussion of the cardiovascular system talking about exactly what the cardiovascular system is. So the organs that make up the cardiovascular system are the heart. The heart's essentially a hydraulic pump. So it's a pump, right, located deep to the sternum. And that pump, when it contracts and relaxes, creates a pressure gradient or creates force that moves blood through a series of tubes that we call blood vessels. So when the ventricles of the heart, which are chambers of the heart we're going to talk about at a later time point, contract, they move blood into blood vessels called arteries. An artery is a blood vessel that carries blood away from the heart. So arteries carry blood away from the heart, and typically in the systemic circulation, you see arteries denoted with red, meaning that they're carrying blood that's oxygen rich or oxygenated. So that blood moves away from the heart, and then those arteries branch and branch and branch many, many times over, and eventually those arteries feed into really small blood vessels called capillaries. Now we have billions upon billions of capillaries. Every cell in your body essentially has an associated capillary network. And so there's lots of them. They're very small and we have lots of them. So the most abundant and most important type of blood vessel is a capillary. And capillaries are so important because that's where exchange takes place. So when you see a blood vessel go from being red to being blue, what that's indicating is that oxygen is being delivered and carbon dioxide is being removed from the tissue. There's an exchange taking place, and the only blood vessel capable of exchange is a blood capillary. Right? That blood is then drained from capillaries and it's returned back to the heart via a system of blood vessels called veins. So veins are defined as blood vessels that bring blood back to the heart. Veins in the systemic circulation, and there are a couple of exceptions to this when we talk about the pulmonary circuit, but veins are typically indicated by blue, which is demonstrating that they're carrying oxygen poor or deoxygenated. I don't like the term deoxygenated. I use oxygen poor more often because deoxygenated is a bit of a misnomer. Your blood is never really deoxygenated, meaning that it's not completely devoid of oxygen, but it becomes oxygen poor, we'll say. So that oxygen poor blood then returns back to the heart where eventually it'll enter a circuit where we deliver it to the lungs to pick up oxygen, get rid of carbon dioxide, and the whole process will begin again. So the heart is a pump. It moves blood throughout the body in a system of vessels known as arteries, capillaries, and veins. And what's moving through those arteries and capillaries and veins is a heterogeneous mixture, a suspension mixture of this fluid called blood. And that's what we're going to be talking about today. The job of the cardiovascular system in a nutshell is essentially to deliver nutrients and oxygens to tissues and to get rid of carbon dioxide and metabolic waste from tissue. So the cardiovascular system is essentially like the highway of the body. It delivers stuff, it transports materials around the body, and it delivers stuff that cells need, and it gets rid of stuff that cells don't need, right? Or that cells need to get rid of. So with that basis, what our focal point today is going to be on is blood. Now I mentioned that blood was a suspension mixture. What that means, if you were to put blood in a tube and you were to insert that tube, either let that tube sit for a long enough period of time or insert that tube into what's called a centrifuge. A centrifuge is a device that spins really, really fast and it separates out the different components of blood using inertial force, right, based on their density. And what you do is what you get at the end of that, if you put blood in a centrifuge, is you see that blood isn't just one red liquid with equally distributed particles. Blood is composed of multiple components. You have the cellular components of blood, you have the blood plasma, and you can separate those out based on their density. So what you'll see separate to the bottom if you put let's say a capillary tube, a little tube of blood into a centrifuge, is at the bottom here because they're the most dense of the elements in blood, the most dense of the components in whole blood, are red blood cells. So down here where it says 42% packed red cell volume, all of these cells down here are red blood cells. Now in general, red blood cells are sacs of hemoglobin, and the job of red blood cells is to physically transport oxygen around the body. Now, these red blood cells deliver oxygen to tissues. They take away some of the carbon dioxide from tissues, so they're gas delivery mechanisms, and that's their job. The focal point for this class will be delivering oxygen to tissues. So when you're pointing this out on your handout, right, in your follow along, those are red blood cells. What are the job of red blood cells? They deliver oxygen to tissues. Now, the cellular components of blood are denser than the liquid component, the blood plasma or the extracellular fluid, so they separate toward the bottom. The most dense of the cells in blood are the red blood cells, and then forming this little layer that accounts for about 1% of whole blood is something called the buffy coat. And that buffy coat is made up predominantly of white blood cells and platelets. Now, white blood cells, also called leukocytes, so red blood cells are also called erythrocytes. Erythro meaning red, cyte meaning cell. White blood cells are also called leukocytes, leuko meaning white, cyte meaning cell. You get this little layer called the buffy coat in this suspension mixture that's been separated into its component elements. And these white blood cells are responsible for mediating immune responses, right? So you can think of them as being key players in the immune system. They target and destroy things like infections. Also in this buffy coat, you find platelets. So about 1% of whole blood is made up of white blood cells and platelets in a healthy individual. And these platelets, also called thrombocytes, thrombo means clot and cyte means cell, are responsible for mediating blood clotting pathways. So if we're going from the bottom to the top here based on density, you have red blood cells, which carry oxygen around the body and deliver oxygen to tissues. You have white blood cells, which are responsible for mediating immune responses. And you have platelets, which play an important role in mediating blood clotting pathways. And you talk about those in detail in A&P 2. This is an intro class. We don't get that much into the blood clotting pathways in this class. All of the cellular elements of blood, meaning all of the elements of blood as an adult or as a child that are made in the red bone marrow, and remember red bone marrow was responsible for making red blood cells, white blood cells, and platelets, all of these cellular elements of blood are referred to as formed elements. So if I say all of the following is a formed element except, or which of the following would be a formed element, your formed elements are your red blood cells, your white blood cells, and your platelets. Now, with the lowest density that accounts for most of the volume of whole blood in a healthy individual, you have what's called blood plasma. Blood plasma, if you've ever seen it, for example, if you've ever donated blood or worked in an apheresis unit, which is a process to separate out blood plasma, again, using a machine called a centrifuge, blood plasma is kind of yellow. And what blood plasma is it's an aqueous mixture, meaning that about 93% of it is water, and then you have a bunch of solutes thrown into that water and mixed up, and it gives this blood, the plasma, kind of a yellowish or kind of a straw-colored appearance. So it's kind of yellow when you ever see it. And what blood plasma is it's the extracellular matrix or extracellular fluid of blood, and it's an aqueous mixture, about 93% water, about 7% solutes. And the types of solutes that you find in blood plasma are nutrients. So you get dissolved nutrients like sugars and amino acids that need to be delivered to cells. You get dissolved chemical messengers like hormones that need to be delivered to cells or taken away from cells, from one cell to another cell. So you also get things like dissolved wastes, like nitrogenous waste or carbon dioxide, right? They dissolve in the blood plasma. So the job of blood plasma essentially, right, that we're going to kind of consolidate it down to or simplify it for this introductory class, is plasma is the component of blood in which nutrients are dissolved. And when you move that blood around the body, nutrients in the blood plasma and chemical messengers are delivered to cells. And then things like metabolic waste, like nitrogenous wastes and carbon dioxide, are then removed from cells, and then they're moved in the blood plasma to the parts of the body that get rid of them, like the lungs, for example, when we exhale. So plasma is where you find dissolved nutrients and chemical signaling molecules that get delivered to cells, and that's also the place where you get rid of metabolic wastes and carbon dioxide. You also find electrolytes dissolved in the plasma. So when you do, for example, a chem panel and they look at the different electrolyte concentrations in your blood, they're predominantly looking at the blood plasma, and those electrolytes have a lot of really important physiological roles. So as you go through and you label this, make sure that you can identify all the different components of whole blood, and make sure that you know what's meant by the term formed elements. Now, hematocrit is a really important physiological measure, right, that tells you about somebody's overall health. And what hematocrit is it's the percentage of whole blood that's made up of just red blood cells. So when you think about the equation for hematocrit, if you were in a class and you got a capillary tube full of blood and you got a ruler, how would you calculate hematocrit? Well, hematocrit is the percent of whole blood that's made up just of red blood cells. So if I was to take my ruler, I'd probably measure the height of the entire column of blood. So let's say I put my ruler next to my capillary tube. I'm not going to measure the clay that's using to plug it up or the empty air. I'm just going to measure blood, and I'm going to get the height of the whole column of blood. So let's pretend that the height of the whole column of blood using a ruler is 100 millimeters. Then I would measure the column of whole blood that's just red blood cells, where you get those packed red blood cells. And let's say that height of that is 42 millimeters. The millimeters cancel, so 42 millimeters divided by 100 millimeters. Millimeters cancel, 42 divided by 100 is 0.42. 0.42 as a decimal is 42%, or you could multiply it by 100 in order to convert the decimal percentage to a percentage. The percentage of whole blood that's just red blood cells is referred to as a hematocrit. So the percent of whole blood that's just red blood cells is referred to as a hematocrit. Now, there are a couple of different abnormalities with hematocrits that tell us something important about a patient's well-being. So the hematocrit for females, and this varies from individual to individual, can be anywhere from 35 to 45%. In men tends to be 38 to like 52%. I'm not going to expect you to remember those numbers because they vary. I'll give you a reference range, right? Hematocrit tends to be a little bit higher in men than it does in women. I would definitely give you the reference ranges, but what I do want you to understand are a couple of different conditions that we can diagnose using the measure of hematocrit. So if we look at this column of blood right here and we see normal blood, right? So we measure the whole column, and then we measure the column that's just red blood cells, and let's say it's 42 over 100 again. 42% is in the reference range for both men and women. Now look at this particular image right here. Now we measure the whole column, which is the same height. Let's say it's 100 millimeters. And then we measure the column that's just composed of red blood cells. And let's say it's 22 millimeters. The hematocrit would now be 22%, meaning the percent of whole blood that's made just of red blood cells is 22%. When the percent of whole blood that's just red blood cells, right, is low, we refer to that condition as being anemia, or an anemic condition. A means without, and the EM, right, pertains to blood. So it's a condition without blood, and even though you have the blood, that's what it actually means when you break down the medical terminology there. So you have anemia, and anemia is a condition in which you have a low hematocrit. Now, if you have a low hematocrit, think about what red blood cells are responsible for, because those will give you kind of the diagnostic criteria that you would expect to see in somebody with anemia, meaning how would they be feeling and how would they appear. So red blood cells are responsible for transporting oxygen around the body. You need oxygen because, as we discussed in a previous lecture, you burn glucose in the presence of oxygen in order to make energy in the form of ATP. If you have a low hematocrit or you're anemic, you're not going to be delivering as much oxygen to tissues. right? If you're not delivering as much oxygen to tissues, those tissues aren't going to be able to generate as much energy, right? It's going to be the limiting factor there. So energy reserves or ATP reserves are going to start to run low. And when you think about somebody who's anemic, what do they look like? How do they feel? Well, typically they complain of fatigue and sleeping all the time. They're lethargic. Their skin can appear pale. And it's a serious condition. There are a lot of things that can lead to anemia. You can have vitamin deficiencies like iron or some of the B vitamins. If you're deficient in those, you can't synthesize red blood cells properly, and that can lead to anemia. You can have bleeding that you're not aware of, let's say along the GI tract, so you're losing a little bit of blood, right? You can have metabolic disorders. There are all sorts of things that can lead to anemia. So you typically see anemia present like that. Women with particularly heavy menstrual cycles, even though most of that menstrual fluid is endometrial tissue, if there's a particularly heavy menstrual flow, right, you can lose a bit of net blood. So there are a lot of conditions that can ultimately lead to you being anemic, right? And it typically presents as fatigue, and it makes sense that it would present as fatigue given what we know about red blood cells. Now, on the flip token, there's polycythemia. Polycythemia is when you have a particularly high percentage of red blood cells, or higher than normal reference range. And with polycythemia, you have a percentage of red blood cells that's elevated comparative to whole blood. So let's say this column is still 100 millimeters and the column of red blood cells is 65 millimeters. Well, now your hematocrit is 65%, and that's a bit high. So when a hematocrit is high, we refer to that as polycythemia. Now, the most common cause of polycythemia in the classes that I teach is dehydration. You lose some water, but not red blood cells, and you see a mild polycythemic condition, meaning that you get a very subtle elevation, right? And you can usually correct that with just drinking water. But there are more insidious causes of polycythemia, like if you've had a splenectomy, meaning that you've had the spleen removed. The spleen plays a really important role in recycling old worn out red blood cells. So if those old worn out red blood cells aren't getting recycled out, it can jack up your hematocrit, right? If you're using medications, so for example, let's say you're undergoing chemotherapy and you need to use a medication like erythropoietin, which is a chemical, it's a signaling molecule that stimulates red blood cell formation. You can get polycythemia. Let's say you're riding in the Tour de France and you want to take you want a blood dope. That can raise your hematocrit. Now, people would artificially raise their hematocrit like competitive athletes because if you have more red blood cells, presumably you're going to be able to carry more oxygen to your tissues and your tissues aren't going to become fatigued as easily. The problem with that is red blood cells are like molasses. So it puts a lot of stress on your cardiovascular system, your kidneys, when you have a consistently elevated hematocrit. So it's something you have to figure out. So there are two different conditions that I'd expect you to be able to diagnose there. Now, with respect to blood, we're going to go over blood plasma first, and then we're going to work to red blood cells, and then we're going to talk about white blood cells. In the blood plasma is an aqueous solution, meaning that it's a water-based solution, about 93% water, with dissolved electrolytes, ions, organic molecules, vitamins and minerals, and some gases, predominantly CO2. Oxygen is not very soluble in blood plasma, so focus on CO2 here. Very, very little oxygen exists in blood plasma. Now, if we focus on this and we focus on the organic molecules, we have amino acids, which are nutrients we need to deliver to tissues so they can make proteins. We have functional proteins, meaning proteins in the blood plasma that serve a specific functional role. That's going to be a big focal point. We have sugars, lipids, wastes. So when you think about blood plasma delivers things like nutrients to tissues and it gets rid of wastes from tissues. That's one of its primary roles. Another primary role of blood plasma is it has functional proteins. And these functional proteins serve specific roles within the body that we're going to go through kind of one by one. So Plasma delivers nutrients to tissues, gets rid of metabolic waste and carbon dioxide from tissues, transports signaling molecules around, and has functional proteins. Our focal point here is going to be on the functional proteins, and we're just going to kind of run through them one by one. So the three functional proteins you need to know for this class are albumins. Now, in general, albumins carry lipid-soluble molecules around, meaning like steroid hormones or little lipid-soluble components, little lipid-soluble signaling factors. They can bind to them, make them soluble in plasma, and physically carry them around. So just like my Toyota Corolla carries me around, right? You could think of me as being like a little lipid-soluble signaling molecule like a hormone, and my Toyota Corolla is being kind of like the albumin. It wraps around me, it takes me right through the streets, which are kind of like the cardiovascular system from point A to point B. And that's one of the functional roles albumins serve. Now, it's not part of your Common Core objectives in Intro to Anatomy and Physiology, but albumins also play a major role in maintaining something called blood colloid osmotic pressure. Now, suffice it to say, that's a really important physiological parameter. It prevents your blood from leaking out of your capillaries and pooling in your tissues. And we don't talk about it much in Intro to A&P, but it's a really important factor. It's also called BCOP. So those are the two primary functions of albumins. Where are albumins synthesized? In the liver. So the liver has hundreds of defined functions. Many of them are synthesizing proteins. Albumins are a class of proteins synthesized in the liver. Globulins, our focal point will be immunoglobulins, aka antibodies, and we'll talk about those when we get into blood typing. And then fibrinogen, which is your body's primary clotting protein. So fibrinogen floats around in your blood, and when the clotting pathway is activated, it's converted to another protein that plays a really important role in blood clotting. So think about that when you get to that particular question on the handout that you're going over, and I think we're still in follow along too. So again, this is an intro class, and we kind of gloss over a lot of what you need to know, but fibrinogen is a protein produced by the liver. Now, fibrinogen is soluble in blood plasma until the blood clotting pathway has been activated. So fibrinogen is floating around just in the blood plasma. And when a blood clotting pathway is activated, meaning you have a cut, for example, in that region, there are chemical factors called clotting cascades that eventually convert fibrinogen into fibrin. Fibrin then is an insoluble protein that essentially forms a net-like network. So it forms these nets in the blood that physically catch blood as it flows through that particular region of the body, which plays a really important role in facilitating the process of blood clotting. So fibrinogen is a really important clotting protein, and a lot of times, like I gave you on your particular handout, doctors will actually take fibrinogen samples, meaning let's say a patient is going into surgery, and that patient says, I'm an alcohol abuser, I'm a chronic alcoholic. And they go, okay, well, we're about to do surgery on this patient. We don't want them to bleed to death on the table. So they take a sample of blood and they'll look at fibrinogen levels to see whether that particular individual is predisposed to a bleed. Now, why would alcohol influence fibrinogen? Well, anything that compromises the function of the liver, right? Like alcohol is going to put stress on the liver. It can damage the liver, and if you damage the liver or tie the liver up with trying to process all this alcohol, it's not going to be able to create adequate amounts of fibrinogen. If fibrinogen levels are really low, that's a serious bleeding risk, right? That presents a serious potential bleeding risk. So that's a value that sometimes doctors get before things like surgeries. So think about that in your handout. Now, when you think about this image right here, we talked about the three different types of blood vessels that we have. So we have arteries, which take blood away from the heart. We have capillaries, which are the point of exchange. They're the most abundant and most important type of blood vessel because that's where exchange between the cardiovascular system and the cells of your tissue occur. And we have veins, which bring blood back to the heart. So what you are looking at here is a capillary, and capillaries are so small that red blood cells go through one at a time. Sometimes they even have to fold in half in order to get through there. Now, even though this isn't drawn to scale, these little geometric shapes that I've thrown in here represent your plasma proteins. Now, what do you notice about the wall of this particular capillary? So blood's moving through this very tiny vessel, and there's pressure behind it. Now, if you had water running through a hose, right, and there were no holes in that hose for water to get out, water would run from one end of the hose to the other. But if you took a nail and poked a bunch of holes in that hose, and then you turned it on, applied a pressure to that water, turned on the faucet, water would start pouring out of that hose. Capillaries are kind of the same. Blood flows in, there's a pressure, right? And those capillaries oftentimes have little holes in the walls of the capillaries, in the membranes of the cell, and this depends on the type of capillary we're talking about, right? We have little holes. So blood pressure is going to force a little bit of that plasma through these holes. These bigger elements like the proteins and the red blood cells can't get through. And the moment that plasma is pushed across the capillary wall and it enters into the interstitial space, meaning that space surrounding cells, the moment that it's no longer in the cardiovascular system, it enters into something called the interstitial space. And the interstitial space is the space surrounding cells. And the moment that plasma exits the cardiovascular system and enters into the space surrounding cells, it's no longer called blood plasma, it's called interstitial fluid. The process by which blood plasma becomes interstitial fluid is called filtration. Why is it called filtration? Because you move the small components in the blood plasma across those little openings or gaps, but these big things get filtered out. They don't enter into the interstitial fluid. Interstitial fluid is then the fluid that bathes the body cells and is the point of exchange. So plasma is delivering good stuff. You have filtration, the movement of plasma across the capillary wall, right into the interstitial space where it becomes interstitial fluid, and then that interstitial fluid exchanges with the cells. Now, some of that interstitial fluid gets reabsorbed back into the blood plasma, so the process by which interstitial fluid becomes blood plasma again is called reabsorption. And whereas good things like oxygen and nutrients and chemical signaling molecules are undergoing filtration and being moved toward the tissues of the cells, the things that are being reabsorbed are things like metabolic wastes and carbon dioxide. So that's how exchange actually takes place in a capillary, right? We have this back and forth movement. Now, I often ask, what do you think you have more of just intuitively? Do you think you have more plasma or interstitial fluid? And people will always say plasma because they're so familiar with it. You actually have much more interstitial fluid. About 2/3 of your extracellular fluid is interstitial fluid. Only about 1/3 is blood plasma, right? So there's much more interstitial fluid than there is blood plasma. Now, when you look at an image like this and you see components of the extracellular fluid consists of all the fluid that exists outside of cells. So one example of extracellular fluid is blood plasma. Another example of extracellular fluid is interstitial fluid. And the final example of extracellular fluid that you need to know is lymph. Now, we get into lymphatic pathways in another lecture, but I just want you to kind of understand what happens at the level of a capillary and how to identify capillaries when you're looking at images. So you have your heart. Your heart contracts. It moves blood into a series of blood vessels called arteries. Arteries move blood away from the heart. Now, our cardiovascular system is a closed system. Blood that goes places needs to return back to the heart. Blood doesn't just, it's not, our entire body isn't bathed in like a coat of blood. It's contained within these tubes that we call blood vessels, and that's what we mean by a closed system. It's one continuous system. So when we deliver blood places, we got to bring that blood back. So the heart beats, blood's moved into arteries. Those arteries branch again and again to form smaller blood vessels called arterioles. Those small arterioles, you can kind of think of them for now as being small arteries, then feed into these guys right here, which are kind of like U-turns in the cardiovascular system. Now we're looking at something very small right now. We're looking at a capillary network. When blood moves into a capillary, first, how can you identify these vessels as being capillaries? These really small vessels that facilitate exchange? They're going from being red to blue. And what that means is that oxygen is being delivered and carbon dioxide is being picked up. We go from having oxygen rich to having oxygen poor blood. The only type of vessel capable of facilitating exchange are the smallest and most abundant type of blood vessels called capillaries. So all these guys in here are capillaries, right? Capillary, blood capillary, blood capillary. So at the level of capillaries, a bit of excess hydrostatic pressure forces plasma across the capillary wall into the space surrounding cells called the interstitial space. The moment that plasma moves from the inside of the blood vessel to the interstitial space, it's no longer called plasma, it's called interstitial fluid. That interstitial fluid bathes those cells, facilitates exchange between those cells, and then whereas we deliver things like nutrients and oxygen to the cells, right? And that process by which plasma becomes interstitial fluid is called filtration. The blood vessel in the capillary, right, at the venous end will reabsorb things like carbon dioxide and metabolic wastes. And then that vein, right, that capillary will eventually drain into a venule, and that venule will form a larger vessel called a vein. Those veins will bring blood back to the heart. So we have our capillaries, and then we have our interstitial fluid. The interstitial fluid, which is much more abundant than blood plasma, is all the fluid around the cells. So if we take a really close look, all of this space right here, if I put a pointer here and said, what type of fluid would you expect to find there? The fluid around the cells in the tissues of your body is called interstitial fluid. And remember, this is a three-dimensional system. It's hard to kind of pick that out here. Now, that interstitial fluid, when it builds up, eventually it'll build up enough pressure to pop open a lymphatic capillary. A lymphatic capillary is not a blood capillary, it's part of the lymphatic system. And the moment that you pop open a lymphatic capillary and that interstitial fluid enters into the lymphatic capillary, it's no longer called interstitial fluid, it's called lymph. That lymph then gets circulated and filtered through lymph nodes and eventually returned back to your blood, where it's called blood plasma again. So you have three types of extracellular fluid. You have blood plasma is extracellular fluid, meaning fluid surrounding cells within the cardiovascular system. Interstitial fluid is extracellular fluid, the most abundant of the extracellular fluid surrounding your cells outside of the cardiovascular system. And excess interstitial fluid gets drained by the lymphatic system and then filtered by lymph nodes before it's returned back to the blood, where it becomes plasma again, and that fluid that flows through the lymphatic system is called lymph. So three types of extracellular fluid circulating all the time: plasma, lymph, and interstitial fluid. Now, within the plasma, we have different cells. We call those cells formed elements. Now, those formed elements are produced via a process called hematopoiesis, which means the formation of blood, right? Poiesis is the formation or generation of, and heme pertains to blood. O is the combining form of the word, so blood making. That's what hematopoiesis means. And what happens during hematopoiesis is you have the stem cells in the red bone marrow are given certain chemical cues where they're told to become either a red blood cell, a platelet, or a white blood cell. So remember, stem cells are cells that haven't differentiated yet. They're cells that haven't decided on what they're going to do. And in order to decide on what they're going to do, they need to be given chemical cues that let them know, oh, it's time to become a red blood cell, or it's time to become a white blood cell. So if I was to show you this pathway and I was to circle these stem cells up here and say, where would you find these stem cells? Well, you find those stem cells in your red bone marrow. And red bone marrow is perfusing all the little nooks and crannies of spongy bone, right? So you find it in spongy bone, that red bone marrow, and the stem cells in red bone marrow are responsible for making red blood cells, white blood cells, and platelets. So all of your formed elements in your blood, your cellular elements, right, whether they be cell fragments or whole cells, are made in your red bone marrow. So these stem cells are actually cells within the red bone marrow. That's why you extract red bone marrow when you're doing like a stem cell transplant, for example. So if we look at the pathway over here, right, If we want to create red blood cells, we need to introduce the chemical factor erythropoietin. And the way EPO is released, and I kind of want you to trace along with me, and you can even look up other YouTube videos on this because we have a limited amount of time and I'm just getting to the main points in your Common Course objectives. When blood oxygen levels drop, so when O2 levels drop, so you're going around and let's say you, for whatever reason, blood oxygen levels drop, like you increase your elevation because you're climbing a mountain. Blood oxygen levels drop. When blood oxygen levels drop, right, that's the stimulus for this particular pathway. So you have O2 poor blood flowing through the kidneys. So that oxygen poor blood flows through the kidneys. And when the kidneys detect there's low levels of O2, the kidneys function kind of like receptors and control centers, meaning they detect the change, meaning the drop in O2, the stimulus, so they detect the drop in O2, the stimulus, and they process that information, and in response they release an effector, which is a chemical signaling molecule called erythropoietin. EPO, which is a hormone essentially, travels through the bloodstream until it gets to the red bone marrow. When EPO enters into the red bone marrow, it binds to receptors on these stem cells and it says, hey, become a red blood cell. Right? When EPO is available, these stem cells differentiate into red blood cells, kick out their nuclei, and turn into these little biconcave discs that we see down here. This particular process is called erythropoiesis. So EPO stimulates the formation of red blood cells. Questions I want you to think about for this upcoming exam is why would athletes who go through things like endurance races, like the Tour de France, for example, why would they use EPO, right, in order to enhance their athletic performance? That's a question for you to think about. And there we go, because that's what blood doping is. Blood doping is essentially artificially elevating your hematocrit. Now, platelets are just cell fragments that are involved in the blood clotting pathway. We're not going to talk too much about the stimulus for the formation of platelets. Suffice it to say, there is a signaling factor there. It's called TPO, or a thrombopoietin. And then we have our colony stimulating factors. Colony stimulating factors are another one I'm going to ask you about, and then I'm going to bring up a clinical scenario in which both of these become important. Colony stimulating factors, there's a specific one for every type of white blood cell, but ultimately the presence of colony stimulating factors will tell these stem cells to turn into white blood cells. And each one has a different CSF. You don't have to know it. So you have these colony stimulating factors that ultimately tell these stem cells to become white blood cells. Colony stimulating factors are released by white blood cells. So let's say you have neutrophils, and those neutrophils are out targeting a bacterial infection, and you have a bunch of bacteria that these neutrophils are gobbling up. The neutrophils themselves will release their specific colony stimulating factor. That colony stimulating factor will say, hey, white blood cell, right? Hey, stem cells, we need more neutrophils. And that will trigger the formation of neutrophils. And that's the same for all the white blood cells. So what triggers the release of colony stimulating factor is the presence of infection or an immune threat, right? And then that colony stimulating factor feeds back and ultimately produces or tells these stem cells to differentiate into white blood cells. Now, EPO and colony stimulating factors were a really important discovery because when you think about how cancer treatment works, we essentially irradiate or we cut. Right? And there are more sophisticated therapies on the horizon. But when you think about how like chemotherapeutics actually function, or radiation actually functions, it functions by decreasing the mitotic activity of cells. So essentially it prevents rapidly dividing cells from dividing. One of the problems with preventing rapidly dividing cells from dividing, like using methotrexate, for example, to kind of dampen down how quickly cells are undergoing cell division, is that cells in your stomach, your stem cells, and your red bone marrow, those are rapidly dividing cells. So before the discovery of EPO and colony-stimulating factors as things we could use clinically, you used to see right cancer patients with the face masks on, assuming that COVID-19 isn't around, and it wasn't because they were worried about giving you an infection; it's because they were worried about getting an infection, right? They tended to be severely anemic, which means low hematocrit, low red blood cells, severely leukopenic, which means low white blood cells. The discovery of EPO and colony stimulating factors, when used appropriately, is actually really important because now we can prevent some of those complications from happening by upregulating the amount of white blood cells and red blood cells that are produced. Now, when you look at this image right here, what you're looking at is red bone marrow. So this is a capillary flowing through red bone marrow. If I gave you a question on the exam that said, which of the following molecules do you think would be elevated in this region of the red bone marrow? Well, we see a process in which a stem cell is becoming a white blood cell, specifically a neutrophil, so it'd have to be colony stimulating factor. As opposed to when we look over here, we see this stem cell maturing into a red blood cell, so it kicks out its nuclei and whoop, comes into the bloodstream. They're essentially sacs of hemoglobin. You'd have to have EPO elevated over here. So that's what's actually happening. You get elevations of these chemicals and they tell those stem cells what to do. Now, when you think about red blood cells, which are going to be our first focal point for today, red blood cells are shaped like little biconcave discs, like these kind of inner tubes or these donuts or these tires, right? That's what biconcave disc is. It means it has this little depression right in the middle right here, and it's a disc with a little depression. They're about 8 micrometers in diameter, and what red blood cells are, for the most part, after they kick out their nuclei, are sacs of a protein called hemoglobin. Now, there are 2 reasons red blood cells are shaped the way they are. So remember that red blood cells carry oxygen around the body. And in order to do that, these red blood cells need to be able to get into capillaries, and capillaries are really small, so this biconcave shape can allow red blood cells to fold, so they can fold as they're moving through capillaries. Another thing about this biconcave shape is it increases the amount of surface area for gas exchange to occur. So that's why you get that little biconcave shape. Now, internally, if we were to look at a section view and we're wondering what fills up a red blood cells are sacs of hemoglobin. They're membrane-bound sacs of hemoglobin. And hemoglobin is a protein, right? So what we're looking at here is what's called the quaternary structure of hemoglobin. We have all these little polypeptide subunits that join together to form the mature protein. That's not the point I want you to remember about this. First off, I've had emails asking me, how does hemoglobin fit into a red blood cell if it's so much bigger? This particular image is not drawn to scale. It's drawn so you can see the structure of hemoglobin. If this was hemoglobin, if hemoglobin was like the size of me, right? So pretend I'm this hemoglobin molecule and I'm standing next to a red blood cell. A red blood cell would be like 10 Empire State Buildings tall, 10 Empire State Buildings wide, and there'd be like 7 million of me in there. So there's millions of these guys that physically fit into the cell, right? We just draw it like this so you can actually see the protein, because remember, when you're talking molecular biology, like proteins, for example, you're talking about things that are super small. So we've blown it up in this image. It's not drawn to scale. And hemoglobin is made up of these globular subunits, and in each subunit we have a heme group. Now, each heme group has a central iron. It's one of the reasons that red blood cells are so dense, because they're sacs of hemoglobin, and each hemoglobin molecule has 4 heme groups, and each heme group has a centerpiece of iron. So it's actually quite dense, meaning that the density of a red blood cell is greater than the density, or it's more dense than like the density of a white blood cell and more dense than plasma, and that's why they sink to the bottom or separate out to the bottom. It's like putting a little iron in a bathtub. It's like a little chunk of iron, it just go bloop and sink to the bottom, right? That's why they have the density they do. Now, when you look at these heme groups, every hemoglobin molecule has 1, 2, 3, 4 heme groups, and each heme group is capable of binding to a molecule of molecular oxygen. So each heme group is capable of carrying a molecule of molecular oxygen. So when you say hemoglobin is 50% saturated, right? or that blood oxygen is 50%, what you're really saying is that if you've ever looked at one of those pulse oximeters and it says, like, let's pretend it said 75% oxygen saturation, to extrapolate on that, even though it's just measuring color, which is not something we're going to get into, right? It's measuring the reflective properties of blood. That would mean that on average, 1, 2, 3 molecules of oxygen were bound to each molecule of hemoglobin for a total of 75% of each hemoglobin molecule, right? Its total oxygen capacity actually being bound to oxygen. So when hemoglobin is 100% saturated, it means each hemoglobin molecule is binding to 4 molecules of molecular oxygen, and where that reversible interaction takes place is at the iron. Right? It's kind of like rusting, but don't worry about that. But that oxygen binds to that iron atom in a reversible way. So if there are 4 heme groups, right, each molecule of hemoglobin, when it's fully saturated, can transport 4 molecules of oxygen. 75% 3, 50% 2, right? So think about those percentages. So now we have the basics of blood. We've talked about how blood cells are formed. And now we're going to talk about one of the key points to our lab today, which is blood typing. And this is going to cap off our discussion of white blood cells before we move into discussing red blood. This is going to cap off our discussion of red blood cells before we move into discussing white blood cells. So we've talked about whole blood, we've talked about plasma, we've talked about a couple of the critical plasma proteins. We brought up an example of one. Then we've talked about the flow of extracellular fluid and the role of blood plasma, right? So we started kind of with our focal point on plasma. Then we shifted to red blood cells. We talked about hematocrit. We've talked about why you would artificially elevate hematocrit doing something like blood doping. And now we're going to talk about blood typing. So we're going to continue our discussion of red blood cells before we get into our discussion of white blood cells. So when you talk about blood type, what you're really talking about is the fact that all of the red blood cells in your body carry little markers on them that signal to the immune system, hey, I'm okay. And that's a simplistic way to put it, but it's the way I'm going to put it now. These little markers you find in the surface of a red blood cell are called blood antigens. So blood antigens are genetically determined markers you find in the surface of a red blood cell. Now, the reason that they're called blood antigens rather than blood markers is because they're capable of eliciting an immune response. So an antigen is just anything that can trigger an immune response. Viruses are antigens, bacteria are antigens, fungi can be antigens, your own tissue can be an antigen. An antigen is just anything that triggers an immune response. A blood antigen is a marker in the surface of the red blood cell that could potentially trigger immune response or an immune response, and that's why we call these markers in the surface of red blood cells blood antigens. So blood antigens are markers embedded within the membrane of red blood cells that are genetically determined and never change. Antibodies are a group of plasma proteins that bind to antigens and do a few different things. They can tag them, they can mark them for destruction, or they can cause them to cluster in a process called agglutination. So when you look at a red blood cell, and we see there are like 600 different blood types, right? But there are only 2 classes that we care about. We care about the ABO group, and we care about the RH group. So when we talk about the ABO blood group, we're talking about the presence or absence of an antigen. Remember, antigens are anything capable of triggering an immune response. A blood antigen is just a marker in the surface of the red blood cell that communicates information to the immune system. So right now we're looking at a red blood cell, and we're looking at just my depiction of a red blood cell. Now with the ABO blood group, right? If I ask you to draw like a picture of a blood cell with somebody who has a certain blood type, focusing on the ABO blood group and the RH blood group. What A means is that they would have the antigenic marker A embedded in the surface of the red blood cell's membrane. What B means is they'd have the antigenic marker B embedded within the surface of the red blood cell membrane. If they had both the A and the B antigenic markers, right, they would be AB. If they didn't have either the A or the B antigenic markers, they would be considered O. So when you talk about ABO blood groups, the A and the B refer to the presence of those markers in the surface of the red blood cell. The O refers to the absence of those markers in the surface of the red blood cell. RH groups are exactly the same. So RH, which stands for rhesus factor because it was discovered in rhesus monkeys, is another little chemical marker in the surface of a red blood cell, another little glycoprotein. And the RH group, right, refers to when you say somebody is positive, that means the RH group is present. And when you say somebody is negative, that means the RH group is absent. So when we look at blood types, right, now we have a marker in the surface. So there are no markers in the surface of the red blood cell at this point. This person, by definition, if they lacked both the A and B marker and the RH marker, they would be O because they have neither the A or the B marker, and they would be negative because they lack the RH marker or the RH antigen in the surface of the red blood cell. Now, even though I'm only putting one on here to emphasize a point, this would really be if you had the A antigen, the A blood antigen, right? They would be all over the surface of the red blood cell. It would be like sprinkling sprinkles on a donut, right? They're all over. They coat the entirety of the red blood cell, but I'm just using one to emphasize a point here. So now we have the A marker in the surface of the red blood cell. This person would now be considered A negative. So they have the A antigen, they don't have the B, they don't have the RH, so they would be A negative. AB negative because now they have both the A and the B marker in the surface of the red blood cell, but they don't have the corresponding RH marker. AB positive. So what you're talking about with your blood types, or when you talk about blood groups and blood types, you're talking about the presence or absence of markers called blood antigens in the surface of the red blood cell. If you lack both A and B, you are O. If you have A or B, you'll be A or B or both AB. The RH marker is just another marker in the surface of the red blood cell. If you don't have it, you're negative. If you have it, you're positive. So when I ask you on your handout to draw a picture of a red blood cell for somebody who's A positive, for example, I'm saying what markers will be in the surface of their red blood cell. So blood antigens are markers in the surface of a red blood cell. They're called antigens because they can potentially trigger an immune response. Antibodies, on the other hand, are not markers in the surface of a red blood cell. Antibodies are molecular pins, right? Antibodies are proteins produced by white blood cells that target infection. So whereas an antigen is something that can trigger an immune response, the antibody is the immune response. So let's say you have an antigen like a virus. White blood cells in your body will start producing antibodies that will pin to that virus and deactivate it. Or a toxin, they could deactivate it or mark it for destruction. So antibodies are these proteins produced by white blood cells that float around and they target specific antigens. Don't mistake or confuse antigen and antibody on the exam. So you have antigens and you have antibodies. Antibodies are proteins produced by white blood cells that can function like little molecular pins that mark cells for destruction. They can deactivate viruses and toxins, or they can function as a glue that clump antigens together, making it easier for the immune system to clean that particular infection out. When an antibody causes the clumping of antigens, that process is called agglutination. So agglutination reactions are really clumping reactions. Now, when you look at this particular image here, what you are looking at here are antibodies, And what these antibodies, these kind of T-shaped proteins that are drawn way bigger than they actually are doing, are they're binding to antigens on the surface of the red blood cell. And they're not causing red blood cells to clot. Clotting is a different reaction. This is not a blood clot. What antigens do is what antibodies do is they bind to antigens, those markers in the surface of the red blood cell. So let's say this was anti-A antibody, and it binds to the A antigen in the surface of the red blood cell. And when they bind to the antigens in the surface of the red blood cell, when they come into contact with one another, they cause the red blood cells to stick together, so it's easier for the immune system to clean them out. When antibodies cause things to stick together or clump together, it's not called clotting. That's a totally separate, right, physiological activity. When antibodies stick to antigens, right? So when the immune system proteins make antigens, which are anything that can trigger an immune reaction, stick together, that process is called agglutination. So what we're going to be looking at today when we go over blood typing is the process of agglutination. And I have another class, so I'm going to have a really nasty, awkward edit. So when antibodies, which are proteins, bind to antigens, they cause those antigens to stick together. So here are red blood cells, and the surface of those red blood cells are these teeny tiny things we call blood antigens. They're like markers in the surface of our red blood cell. And what these antibodies do, these proteins, is they bind to one, they bind to another, and when they come into contact with one another, they stick and they begin to clump. It's not a blood clot, It's called an agglutination reaction when immune proteins called antibodies cause antigens to clump. So when you're looking at your blood surface antigens and blood groups, we care most about the ABO blood group and the RH blood group. The example you're getting up here is focusing the attention on the ABO blood group. So if you have neither antigen A or antigen B, right, your blood type here would be O. So there would be O, right? Because you have neither A or B antigens in the surface of your red blood cell. Now, your body will naturally produce antibodies to the antigens that you don't have. So if you have this red blood cell here, by the way, this would be O negative because we don't see the RH antigen there either, right? Your body will naturally start to produce anti-A antibodies and anti-B antibodies. So the antigen is the marker in the surface of the red blood cell that can potentially trigger an immune response, and the antibody is what binds to that particular marker, causing agglutination, which is the immune response. These proteins are produced by white blood cells. The markers in the surface of the red blood cell are genetically determined at birth, right? They don't change. Antibodies, on the other hand, can, but we're not going to talk a lot about that in this class. So you always produce the antibody for the antigen that you don't actually have in the surface of your red blood cell. So here we have neither A or B antigens. So this person, if I was to ask you which antibodies would be in this person's blood, they'd have both anti-A and anti-B antibodies. You produce the antibodies for the antigens you don't have. If this person had an A antigen and they produced an A antibody, that A antibody would bind to the antigen, cause the red blood cells to agglutinate, and that would produce a transfusion reaction. It would essentially kill yourself. You would never produce the immune response to an antigen that you had, only to one that you don't have. So if you have neither A or B antigens, you're going to produce both A and B antibodies. So somebody with type O negative blood will produce both A and B antibodies. And this becomes really important in understanding transfusion reactions, who you can give blood to and who you can't. So if you have antigens A and B in the surface of your red blood cell, you'd be AB, and in this case, AB negative. Now, we're not going to talk about Rh antibodies in this class, right? But If you produce both A and B, or if you have both A and B antigens in the surface of your red blood cell, you're not going to produce either anti-A or anti-B antibodies. Why? Well, because those antibodies would, right, attach to these, cause them to clump and trigger an immune response, and you don't want to be destroying your own red blood cells. So somebody who's type AB would produce neither anti-A or anti-B antibodies. Somebody who's type B wouldn't produce anti-B antibodies because they have the antigen and you don't want that antibody targeting the antigen. They'd produce anti-A antibodies. Same thing with somebody who has antigen A in the surface of their red blood cell. They would produce anti-B antibodies, but not anti-A. So these are the antibodies that correlate with the blood types above. What do you find in this person's blood? And those are going to be questions that we hit really hard. So what we're going to be using today is what's called an Eldon card. Now, an Eldon card, if you were to get the actual card in a lab, looks kind of like this with the different colored solutions on each well, right? We'll call these Eldon wells. And you'd write your name, and what you would do is you would activate this solution with a drop of water. And essentially, it's dry before you put the drop of water on, and you want to get that stuff up into the water. What you find on the Eldon card are antibodies. They're not your body's antibodies, right? So a lot of people will say, well, wait, I thought we wouldn't produce antibodies. They're not your body's antibodies. They're antibodies that have been produced in a lab and put on the card. So they're not your antibodies. They're artificial antibodies on the card, right? So in this well, you have anti-A antibodies. In this well, you have anti-B antibodies. In this well, you have anti-RH / or AKA anti-D antibodies. And in your control, you have no antibodies because if the control ever clumps, you just have a card that is no longer working. So what I'm going to show you are images like this, right? So I'm going to show you images like this. And what we're going to talk about is based on the agglutination reaction, what would the blood type of the person be? What antigens would they have in the surface of their red blood cell, right? What antibodies would they produce and who could they donate to? So we're going to call these we're going to call these no clumping or no agglutination, and we're going to call these where you see the little dots clumping or agglutination. So we got no agglutination, no agglutination, agglutination. When you see agglutination, that means that an antibody has bound to an antigen. So the antibodies on the card have bound to the antigen and they're causing the red blood cells to clump together. So what we're going to be looking at are agglutination reactions. Oh, there are some awkward edits in there because I'm getting calls and emails and things. COVID-19, thanks, buddy. So, we know what agglutination reactions are. They're when antibodies bind to antigens and they cause them to clump together. Not a blood clot, an agglutination reaction. Very different. When we're looking at agglutination reactions in the lab, we're going to use something called an Eldon card. So this was an example of an Eldon card that you'd get in the actual lab. And what you see are these different colored substances on the Eldon card before you even put blood on the Eldon card. So here we have anti-A, and what that means is anti-A antibody. So what's on the Eldon card before you introduce blood onto the Eldon card is antibody. It's not your body's naturally produced antibodies. They're artificially lab manufactured antibodies, right? So they've put antibodies on the Eldon card in order to evaluate what antigens a person has in the surface of their red blood cell. So we use these antibodies to probe for the presence of antigens. So remember, the blood antigens are the markers that stick out of the surface of the red blood cell. The antibodies are the immunological proteins that bind to those markers and ultimately cause them to clump together, which is what an immune response to a blood antigen would be. So you have anti-A antibody, anti-B, anti-D, which also means anti-RH antibody, and then you have a control with no antibodies. And the question reads, based on the agglutination reaction of the Eldon card below, so agglutination reaction of the Eldon card above or below or wherever it is in the question, what would this person's blood type be? Well, we're going to say when blood clumps together on an Eldon card, we're going to represent it on a diagram as being kind of these dots, right? So that represents red blood cells clumping together, and that represents a positive agglutination reaction. When there's no agglutination reaction, we're just going to represent it as a solid red, and that represents a negative agglutination reaction. And what we're going to do in this slide is we're going to figure out exactly what these agglutination reactions tells us about that patient's blood. So we have Billy and Sam. I say, okay guys, put your water drops on each of your wells. So Billy puts a drop of water on the anti-A well, the anti-B well, the anti-RH well, and the control in order to bring those antibodies into solution so they can actually interact with the antigens on the surface of the red blood cell. So we put our drops of water on, and then we put our blood on. And Billy's agglutination reactions look like this. So you put a drop of blood into the anti-A well, and there's no agglutination. What that means is this anti-A antibody doesn't have anything to bind to, and the only thing the anti-A antibody binds to is the A antigen. So what you've just done, right, with this negative agglutination reaction is you've ruled out that Billy can have A antigens in the surface of his red blood cell. So definitely no A antigens. No agglutination, no B antigens, because this anti-B antibody doesn't have anything to latch onto. No agglutination, no RH antigens, because this anti-RH antibody doesn't have any of the surface markers to latch onto. And then the control should never clump. And I won't give you well, maybe I could be tricky and give you a clumped control. That would just mean the card is garbage and the test is useless. So we do Sam. Well, when Sam introduces his blood into the wells, you see that the red blood cells clump, and you get this very definitive kind of grainy or clumping pattern. So what we see here is a positive agglutination reaction. What that means is the anti-A antibody bound to the A antigen in the surface of the red blood cell, causing the red blood cells to stick together. Same thing with the anti-B antibody. We get agglutination, the anti-B antibody bound, right, to the antigens in the surface of the red blood cell, causing them to stick together. Same thing with the anti-RH, and then the control, you should never have an agglutination reaction. So if you want to pause and see whether you really understand where we're going to go with the next step, you can do that. But let's say my first question to you is based on the agglutination reactions above, draw a picture of Billy and Sam's cells. So how would you draw a picture of Billy's cell? Well, or Sam's cell, or Billy's cell. Well, Billy had no agglutination across the board, meaning these antibodies didn't have anything to bind to, right? These antibodies were just floating around, but there was nothing that they could interact with. Because of that, we had a negative agglutination reaction on each one of the wells, meaning that there were no antigens, right? There were no antigens that corresponded with these antibodies. So you know he doesn't have antigen A, antigen B, or antigen RH in the surface of his red blood cell, and if he lacks all of those antigens, Billy would not have anything popping out of the surface of his red blood cell that we're concerned with for this particular exam, right? So that would be a picture of Billy's red blood cell, no antigens. Sam's red blood cell, on the other hand, because when you introduce the blood to anti-A antibody, the antibody clung to the antigen, causing the blood cells to clump together or agglutinate. That's agglutination positive, right? It would mean that he would have to have the A antigen in the surface of his red blood cell. So now we draw the little A. Now, are there just 3 of them? No, there are thousands. It's like sprinkles on a donut, but this is an artist's depiction. Right? So then I see the agglutination with the anti-B. It means the anti-B antibody bound to the B antigen, causing agglutination. So they have to have the B antigen in the surface of the red blood cell. Same with the RH. So when we start asking questions, if I ask you to draw a picture of the cell, I'm saying, do you understand, based on an Eldon card agglutination reaction, what blood type the individual, or what antigens would be in the surface of that person's red blood cell? And based on the antigens, the fact that this person has A, B, and RH antigens, do you know their blood type? Well, yeah, they would be A, B positive, because they have both A and B antigens and because the Rh is present rather than absent. So this person would have AB positive blood. The antigens in the surface of the red blood cell would be antigen A, antigen B, and antigen Rh. What antibodies would this person produce naturally? Not on the card. The card is not the person. The card is artificial antibodies, and we're asking what antibodies would this person produce? They wouldn't produce any. Because if they did produce anti-A or anti-B or anti-Rh antibodies, they would destroy their own blood and cause it to clump up in their cardiovascular system, right? So now we do the same with Billy. We say, what blood type is Billy? Well, he's O neg. He doesn't have any antigens in the surface of his red blood cell. We say antigens, none. But if Billy doesn't have any antigens, he will produce antibodies. He'll produce anti-A and anti-B antibodies, right? These aren't the antibodies on the card. These are the antibodies being produced in Billy's bloodstream. So now we say, well, what about, right, donor-recipient status? Can Billy donate to Sam? Can Sam donate to Billy? I'm pretty lost. So you go, okay, now that we've determined the antigens and the antibodies present in the presence of these in the person's red blood cell membrane, now we can start determining donor-recipient status, right? Is that a no-go, or can that donor-recipient status occur with no negative interactions? So can Billy donate to Sam? What you always ask yourself with donor-recipient status is you look at the donor antigens. So you look at the donor antigens. Those are the only things that matter on the donor, are the antigens in the surface of the red blood cell. And you look at the recipient antibodies. If the donor antigen ever corresponds to the recipient antibody, meaning that the markers in the surface of this red blood cell are going to interact with the antibodies in the presence of the recipient's blood, that is a no-go. You can't do that, right? So let's look at this one. There are no antigens, and this person produces no antibodies. So Billy is O negative. Can he donate to Sam? Yes. He doesn't have any antigens to trigger an immune response. And Sam doesn't have any antibodies to facilitate an immune response. So you can't trigger an immune response, and there are no immune antibodies there. Yeah, Billy can donate to Sam. In fact, because Billy has no antigens in the surface of his red blood cell, being O negative, Billy is considered the universal donor, right? So Billy can donate to Sam, sure, but can Sam donate to Billy? So now we look at the antigens in the surface of the red blood cell, A, B, Rh antigens, and we look at the antibodies Billy produces. So if you were to introduce Sam's blood into Billy, the anti-A antibodies would bind to the A antigens, causing the blood to clump. The anti-B antibodies would bind to the B antigens, causing the blood to clump. You would get a massive agglutination reaction, and then eventually all those red blood cells would burst open, and you would kill Billy if you introduced Sam's blood into Billy. So no, Sam cannot donate to Billy. In fact, Sam can only donate to other people who are AB positive. So because Sam doesn't have any antibodies, meaning he's not going to trigger an immune response to people who donate to him, right? AB positive is considered the universal recipient. But he can only donate to other people who are AB positive, right? Multiple times, but we won't elaborate on that. So that's what you're looking at with donor and recipient status. You always look at the donor antigens and the recipient antibodies. Whereas O neg is the universal donor because there are no antigens. AB positive is the universal recipient because there are no antibodies. What would you rather be? Probably AB positive because we're all a bunch of selfish jerks. So if you were to give somebody, right, blood in which the red blood cells have an antigen to which they have a corresponding antibody, what you'll trigger is a transfusion reaction. And transfusion reactions can be quite serious, right? Those red blood cells get popped open eventually after they clump. That hemoglobin clogs up the kidney. It shuts the kidney down. Your blood pressure drops. You're not getting blood to areas like your brain. Your immune system activates. If you're fevered and chilled, your heart starts going crazy. And you can eventually die. So blood groups, blood typing, blood antigens, and donor recipient status is a really important part of nursing training. So if you want to pause for a minute and figure out this particular Eldon card, you can. What I'll tell you here is that all the little grains of sand, right? All the little grainy looking things here, the little blood agglutinated clumps of red blood cell that look like grains of sand, those are agglutination. This is no agglutination. That is definitely agglutination, and then the control should never agglutinate. See whether you can answer all those questions, right? Can you draw a picture of that red blood cell? Can you tell me who that person can donate to, who they can't donate to? Can you tell me that person's blood type? What antigens are present on their red blood cell? What antibodies you would find floating around in this patient's blood? Not on the card, but in the patient's blood who put their blood on the card. So now we've gotten through blood typing. The final portion of this class is going to be over white blood cells. So about 1% of whole blood, which is made up of red blood cells, white blood cells, platelets, and blood plasma, about 1% of that whole blood is made up of white blood cells. And each of the respective white blood cells exists in a different ratio, meaning that some are more abundant than others. Now, if you look at the basic designation of white blood cells, you get granulocytes and agranulocytes. These granulocytes are called such because when you stain them and put them under a light microscope, they have these very definitive grains. Notice that they get the suffix "phils" after philos for love. So neutrophils love neutral stain. Eosinophils, eosin is an acidic stain, so they love acidic stains, which are always really red. And basophils love basic stains, which are always really purple, dark blue. On the other hand, the agranulocytes, where you don't see those really big granules, even though there are granules in their cytoplasm, but don't worry about it, where you don't see those big granules, we call them agranulocytes. And notice that they end with cyte, so we have lymphocytes and monocytes. And of the 1% of whole blood that's white blood cells, there's a different ratio of these white blood cells, meaning some are more abundant than others within the blood. And there's a mnemonic for remembering that ratio. It's called "Never Let Monkeys Eat Bananas." So at any given point in time, you should have the most neutrophils, followed by lymphocytes, followed by monocytes, followed by eosinophils, followed by basophils. Now, one of the things I would have had you do in the lab is what's called the differential white blood cell count. So I would have had you take your blood, smear it on a piece of glass, put it in some stain to stain those white blood cells, and then count those white blood cells to do a differential white blood cell count. Now, when you do a differential white blood cell count, essentially you're counting the total number of white blood cells, and you usually do it on a slide that has a grid, and you count 100 white blood cells, right? And then you count the number of white blood cells that are neutrophils, lymphocytes, monocytes, eosinophils, and basophils, and you divide the total number, for example, of neutrophils by the total number of all white blood cells to get a percent of the white blood cells that are neutrophils. And that's called the differential white blood cell count. Differential white blood cell counts are a common diagnostic because if you have some white blood cells that are elevated, it can tell you something about what that person or a condition that person may be suffering from. So let's look at our first type of white blood cell. Neutrophils, body's most abundant type of white blood cell. If you look at the cytoplasm, it looks kind of like a purpley blue stain, but that's really an intermediate between the acidic and the basic stain. It's kind of neutral. So it's not super purple and it's not super red, so we get those neutral granules. But what I think is the more telltale sign of a neutrophil is what's called a multilobar nuclei. So this isn't multiple nuclei. It's not a multinucleated cell. This is a nuclei that deforms, and the nuclear envelope forms definitive regions within the cell that we call lobes. So it's not a multinucleated cell. No, it is a multilobar nuclei. So neutrophils are big. They have these multilobar nuclei, this kind of relatively neutral stain. And what neutrophils do is they're your body's first line of defense against bacterial infection. Neutrophils go around and they physically actually gobble up bacteria. So if you want to see how a neutrophil works, this is a little Staphylococcus gonorrhea bacteria here. And this is a white blood cell called a neutrophil. And what it does with that bacteria is it tracks it and then it comes up and gobbles it. And that's how a neutrophil actually destroys the bacterial pathogens it's targeting. If you were to do a differential white blood cell count and you were to see that neutrophils should be between 40 and 60% of all of the red or all of the white blood cells in that you've counted, right? That would be a reference range for them. And you see that neutrophils account for like 75% of the white blood cells. You may start to ask yourself, well, the body would only do that, would only upregulate the number of neutrophils, meaning make more neutrophils in the presence of a bacterial infection. So you got a patient coming in and they've had a cough for a couple of months and they can't get rid of it. You do the differential white blood cell count. You see that neutrophils are elevated. That's a pretty strong indicator that that may be a bacterial infection. And what you can do at that point is you can go, okay, that might be a bacterial infection. Let's prescribe an antibiotic. Because antibiotics aren't effective against fungi unless you, well, I won't get into that metronidazole anyway. That the antibiotics are effective against bacteria and only bacteria, right? So if you give it for a viral infection, you may actually have a counterproductive effect by wiping out your good bacteria in your gut. So the differential white blood cell count is important. Now, eosinophils, when you look at their cytoplasm, it's really grainy and red. Really red. They also have these bilobar nuclei, 2 defined lobes, but the cytoplasm is really grainy and red. And where you see eosinophils elevated is whenever the body is targeting parasitic infections. So things like tapeworms, blood flukes, malaria, amoebas, those are parasites. And parasites have the same cellular machinery we do, meaning that they have membrane-bound organelles within their cells. So it's really hard to target parasites with medications. Antiparasitic medications can be pretty hard on you. So you want to make sure before you give somebody a really harsh medication that, in fact, that person is suffering from a parasitic infection. Now, if you look at eosinophils in vivo, this is a little blood fluke that we're looking at right here, and these are all eosinophils migrating over and essentially stabbing it with little proteins that are causing the cells in the fluke to lyse, to burst open, and eventually it'll kill the fluke. So when you see eosinophils that are elevated, right, it's probably indicative of something like a parasitic infection. And again, there's a mechanism there, right? You see them just swarming this thing. So an antiparasitic might be an appropriate course of treatment, but you don't tread into that lightly. Now, basophils. Basophils have these bilobar nuclei. They're big cells. Look at their size comparative to those red blood cells. And they have this really dark to purple staining granules in their cytoplasm. Now, what basophils do is they mediate histaminergic responses, meaning that in response to something that triggers the immune system, they start producing histamine, which promotes inflammation. Now, inflammation can be good, right? Or inflammation can be bad if it's not warranted. So one of the things I always find so interesting is when you get a blood test to see if you're sensitive to, like, let's say, a food, right? And they're doing a blood allergen test. What they'll do is they will isolate your basophils in a solution, right? The lab technician in this lab experiment, and they will introduce the thing they think you're allergic to. Like, let's say you say I'm allergic to cedar pollen. They'll put cedar pollen in the solution. So let's pretend these are basophils, and before the cedar pollen has been introduced, they're not doing anything. You put in the cedar pollen, they start degranulating. And all of those little granules are histamine, and histamine is going to dilate capillaries and, cause the migration. It's going to trigger everything associated with inflammation. So if you see basophils elevated, right, that's a pretty strong indicator that it's probably some kind of inflammatory response or allergic reaction, right? Lymphocytes. Lymphocytes are the body's primary defense against viral invaders. It's also the body's primary defense against opportunistic cancers. So lymphocytes are really, really important. A lot of times when we talk about the immune system, we are talking almost exclusively in many respects about what lymphocytes do, because they're the only type of white blood cell that can target viral infections, cancerous cells, and they're the only type of white blood cell that can form what's called a memory, right? Meaning that if they fight one infection one time, they'll create a bunch of cells that float around in your blood and remember that infection for future infections. So you can't become infected with that particular pathogen again. Or if you do, the body launches a massive immune response and you'll probably not even know it. So when we think about things like vaccinations, what they're tweaking is the activity of these lymphocytes. So lymphocytes have this gigantic kind of spherical nuclei with this very small cytoplasm halo. But they are a really important type of white blood cell, and if you get an elevation in your lymphocytes, it could indicate a few things. It could indicate like a viral infection. It could also indicate something like cancer, but it's more likely that it'll indicate a viral infection, and then you can start looking at other things to distinguish between them. So when we talk about lymphocytes, B lymphocytes, you're going to hear the term B lymphocyte. I wouldn't expect you to distinguish those histologically, but they work sequentially. So the first thing B lymphocytes do is they're going around and they're looking at things in the body, right? And they see a little viral invader like coronavirus. They bind to it and that triggers these lymphocytes. So these lymphocytes get angry and then they start creating more of themselves, more and more B lymphocytes, right? They'll even create memory B lymphocytes, which just hang around in case you ever come into contact with this COVID-19 here again. So that's a virus right there, and these lymphocytes come into contact with it, and they start replicating like crazy. And they produce a little subset of cells called memory cells. It's what's called adaptive immunity. And we don't get into that much in this class, but you form, some of them will just stick around just waiting for this guy to show up again. So you get them and they become activated, and once they become activated, they start cranking out from the rough endoplasmic reticulum these Y-shaped proteins called antibodies. So when you're thinking about where antibodies come from in the body, right, why you actually find them in the blood, it's because these B lymphocytes are cranking out antibodies. Now, what do antibodies do? They can bind to and they can neutralize a viral infection like they've done here, or they can bind to and they can cause antigens like blood antigens or anything that triggers an immune response to agglutinate or clump together. T lymphocytes, on the other hand, carry out what's called cell-mediated immunity, and they recognize specific, meaning they have adaptive responses, they recognize specific cells, but they don't work by producing antibodies. What T lymphocytes do is they actually come and they poke little holes in whatever cell is infected. Like let's say this cell was infected with a virus. Let's say that this was a cell in your respiratory tract infected with the coronavirus. T lymphocyte will come over and kill that cell. So it doesn't release antibodies. It'll look at what cells have been pinned by antibodies and it'll come over and it'll kill them. And what you're looking at there is a T lymphocyte killing that cell that's been infected with the virus. So one prevents deactivates the virus or causes the virus to clump, right? Or pins the cell and says, hey, I'm infected with the virus. The other type of lymphocyte just kills any cell that's been infected with the virus or a cell that's become cancerous, right? Now, monocytes are just big daddy cells that function as macrophages. They're huge. They have this kidney bean-shaped nuclei and this kind of clear cytoplasm. And what they do is they're a cleanup crew. They just come around and they gobble up anything, dirt, debris, pathogens that have been tagged with antibodies, and they just come around and gobble things up. So they can be elevated in response to a number of different infections. And they're huge. They're just garbage trucks, man. They just come through and get rid of stuff. So when you look at this depiction of a macrophage or a monocyte here, what it's doing is it's just migrating into this region and just grabbing all these bacteria and going gobble, gobble, gobble. Just gobbles them up. It's huge, man. They're huge cells. I mean, look at the size of that comparative to a red blood cell. So that's it. Have a good one. I'll make 15 more of these and you'll enjoy them as much as you enjoyed this one.